For decades, the accepted explanation was simple. The Basques were Europe's last Paleolithic survivors, genetically sealed off from every major migration that reshaped the continent. DNA proved that wrong. Bronze Age steppe migrants replaced nearly all male lineages across Iberia, and the Basques were no exception. They carry the same R1b paternal haplogroup found wherever Indo-European languages spread, yet they never adopted one. Their maternal lines, meanwhile, held remarkably steady. So if the Basques absorbed the same demographic upheaval as everyone else, what actually made them distinct? Euskara stands alone in Europe. It is the only language on the continent that cannot be connected to any known language family. Linguists have spent over a century searching for relatives, comparing Basque roots to Indo-European, Uralic, even Afro-Asiatic languages, without finding a single consistent cognate. Attempts to link Euskara to ancient Iberian or Aquitanian have failed under systematic comparison. No shared core vocabulary, no matching grammatical structures, and no regular sound changes tie Basque to its neighbors. This linguistic isolation is so complete that Euskara is classified as a true language isolate, not a branch or a cousin, but a survivor with no known siblings. The mystery of Euskara's origins has long puzzled scholars. Its basic words for body parts, family, and counting bear no resemblance to Spanish, French, or any Indo-European tongue. Even computational algorithms designed to find hidden links between languages return no credible match for Basque. The language's unique vigesimal base, 20 counting system, and distinct syntax reinforce its separation from the Romance world that surrounds it. Roman occupation and later migrations left only a handful of Latin loanwords in Basque, mostly for modern concepts, but the core structure remained untouched. This linguistic anomaly drew early researchers to wonder if the Basques were biological outliers as well. If their language was so distinct, perhaps their genes were too. By the mid-20th century, blood group surveys began to reveal unusual patterns among Basque populations. But the language mystery came first. Euskara's stubborn independence set the stage for decades of biological inquiry, as scientists tried to explain how a language could survive so long without relatives, and whether that survival hinted at deep genetic separation. The search for an answer would soon move from dictionaries and grammar tables to blood samples and eventually to the genome itself. Blood group surveys in the 1940s provided the first hard biological evidence that the Basques stood apart from their neighbors. Medical teams working in remote Pyrenean valleys and Basque diaspora communities collected blood samples from thousands of volunteers. Their results were unexpected. Nearly half of Basque individuals tested Rh negative, compared to less than 15% in nearby Spanish and French populations. This anomaly was not limited to a single village or province. Across 18 microregions, the frequency of Rh negative blood ranged from about 30% in the coastal west to 47% in the inland valleys of Navarre and Sul. The ABO blood group data revealed a parallel pattern, with type O exceeding 50% in some districts, again far above the European average. This serological profile raised immediate questions. The Rh negative allele, which results from a deletion at the RHD locus, is rare elsewhere in Europe and almost absent in Asia and Africa. Early investigators speculated that such a high frequency could only result from a founder effect or prolonged genetic isolation. Statistical analysis of the blood group data supported this, showing that the Basques were an outlier, even when compared to other populations with limited gene flow. Researchers in the 1940s and 1950s debated whether the Basques represented a Paleolithic relict, a population that had survived in isolation since the last ice age. The blood group data seemed to support this idea, especially when combined with the linguistic evidence. However, the lack of molecular tools meant these early surveys could not distinguish between ancient continuity and more recent demographic events, amplified by isolation and genetic drift. By the 1970s, the Rh negative puzzle had become a cornerstone of Basque research. Theories multiplied. Some argued for selective advantages, others for repeated bottlenecks in small valley populations, but all agreed that the Basques' blood group profile was not a trivial accident. It was a quantitative marker of distinctiveness, one that demanded explanation beyond language or geography. The stage was set for geneticists to dig deeper, using emerging molecular techniques to clarify whether the Basque anomaly reflected true ancient separation or the aftershocks of population history. For decades, two rival theories shaped the debate over Basque origins. The first, often called the Paleolithic relict model, argued that the Basques were direct descendants of Western European hunter-gatherers who survived the last ice age in isolation. This view predicted that Basque genomes would show uninterrupted continuity with Mesolithic and early Neolithic populations, preserving ancient maternal and paternal lineages without major foreign input. Under this model, genome-wide surveys would reveal high proportions of Western hunter-gatherer ancestry, a scarcity of steppe or Near Eastern genetic signatures, and deep divergence from neighboring populations. The competing hypothesis, sometimes labeled the exotic migration model, suggested that the Basques descended from a later wave of migrants, possibly from the Near East or North Africa, who entered Iberia after the Neolithic. According to this scenario, Basque DNA would carry signals of external ancestry not present in pre-Neolithic Iberians. Genome-wide data would be expected to show clear markers of Near Eastern or Mediterranean admixture, and the Basque gene pool would be more closely aligned with populations from those regions than with ancient local groups. Each theory set distinct expectations for the genetic record. The Paleolithic relic model anticipated a strong match between Basque genomes and those of ancient hunter-gatherers, with little evidence of steppe-related or later admixture. The migration model predicted a break in continuity, with Basques showing ancestry components absent from early Iberian samples. Both models assumed that genetic distinctiveness could be traced to events before the Bronze Age, either through survival or replacement, but neither accounted for the possibility that later demographic shifts might have played a decisive role. Historians and anthropologists in the 20th century debated these models using linguistic, archaeological, and blood group data, but lacked the means to resolve them. Without ancient DNA, it was impossible to distinguish between ancient continuity and later replacement. The arrival of genome-wide sequencing offered the first real test, allowing researchers to compare ancient and modern samples directly and to track the timing and sources of admixture. The central question became not just whether the Basques were different, but when and how that difference arose in the context of Iberia's complex population history. Between 2600 BCE and 2400 BCE, the genetic landscape of Iberia underwent a profound transformation. Genome-wide analysis by the Olalde Research Group, published in 2019, revealed the arrival of steppe ancestry linked to the Pontic-Caspian region. This migration introduced a new genetic component that spread rapidly across the peninsula. Isotope analysis of ancient remains confirmed that these newcomers were not local, their origins traced northeast, far beyond the Pyrenees. The demographic impact was sweeping. Steppe-related Y chromosome lineages, especially R1b M269, rose from near absence to dominance within a few centuries. Bronze Age male burials show this replacement clearly, with indigenous paternal lines nearly disappearing. In contrast, mitochondrial DNA, passed down maternally, retained continuity with earlier Neolithic and hunter-gatherer populations. This pattern points to a sex-biased admixture event where incoming males intermarried with local women. Archaeological evidence aligns with the genetic data, with new burial customs, metalwork, and settlement patterns appearing alongside the steppe signal. Across much of Iberia, the steppe ancestry fraction in genome-wide data rose to between 20 and 40% by the end of the Bronze Age. These findings overturned the older assumption that the Basques and their neighbors were untouched descendants of Paleolithic hunter-gatherers. The Olalde 2019 study established that the Basques too carry steppe ancestry, though at lower levels than other Iberians. The real distinction lies not in the initial impact of the Bronze Age migrations, but in what happened next. While the rest of Iberia experienced further waves of admixture during the Iron Age, and later, the Basque region became increasingly isolated. The steppe ancestry that arrived with the Bronze Age newcomers remains detectable in Basque genomes today, but the subsequent demographic upheavals that reshaped the peninsula largely bypassed them. This continental scale shift set the background against which Basque genetic distinctiveness would later be defined, not by total avoidance of admixture, but by the preservation of an ancient genetic layer as others faded. In 2021, a team led by Flores-Bello and Comas published the most detailed genetic study of the Basque population to date. Their dataset included 1,970 individuals sampled from across the Basque country, Navarre, and the French Basque provinces, with each participant genotyped at over 629,000 markers. This dense micro-regional approach allowed the researchers to test whether genetic distinctiveness was uniform or varied by valley, dialect, and proximity to historical borders. Principal component analysis placed modern Basques in a tight cluster that overlapped almost exactly with Iron Age genomes from the same region. In contrast, Neighboring Spanish and French populations showed pronounced shifts away from this cluster, reflecting centuries of additional gene flow from the Mediterranean and North Africa. Statistic tests confirmed that the genetic distance between Basques and their nearest neighbors was greater than the distance between many other European populations, separated by hundreds of kilometers. The study found that the Basque genetic profile is best explained by continuity from an Iron Age population that already carried a limited proportion of steppe ancestry. Modern Basques retain about 10% steppe-derived DNA, a figure nearly identical to Iron Age individuals from Cantabria and Navarre. In surrounding regions, the steppe component rises to 20% or higher, reflecting later admixture events linked to Roman, Visigothic, and Moorish movements. Yet, in the Basque valleys, genetic influx after the Iron Age is negligible, with no detectable signal from these later migrations. The researchers used QP-ADM and QP-graph modeling to test for hidden admixture pulses, but the best fit models required no substantial gene flow after 500 BCE. The overlap in ancestry proportions, supported by narrow confidence intervals, points to a scenario of demographic stasis in the Basque core, while the rest of Iberia continued to mix. Fine-scale analysis revealed subtle genetic differences between valleys and dialect zones, but these microclusters all shared the same Iron Age foundation. The persistence of R1b M269 Y chromosome lineages introduced during the Bronze Age coexists with a stable maternal pool dominated by haplogroup H. This contrast, male lineage turnover paired with maternal continuity, mirrors the broader pattern seen across Western Europe, but is preserved in unusually pure form among the Basques. The Flores-Bello study resolved a decades-long debate. Basque distinctiveness is not a relic of Paleolithic isolation or a product of unique ancestry, but the outcome of early Bronze Age admixture followed by 2.5 millennia of genetic isolation. The genetic wall that formed after the Iron Age remains visible in the data, separating the Basque valleys from a continent in flux. Genome-wide ancient DNA has forced a fundamental revision of Basque population history. The old models, which framed the Basques as untouched descendants of Paleolithic hunter-gatherers or as a population shaped by a unique migration, cannot account for the full pattern revealed by dense genomic data. Principal component analyses and admixture modeling show that Basques share the major demographic events that shaped the rest of Iberia up to the Iron Age, including the arrival of steppe ancestry and the near-total replacement of Y chromosome lineages by R1b M269 during the Bronze Age. However, the defining feature of the Basque genetic profile is what happened afterward. While surrounding regions absorbed successive waves of gene flow from the Mediterranean, North Africa, and later European sources, the Basque valleys remained insulated. Genome-wide data from both ancient and modern samples demonstrate that gene flow into the Basque region slowed to negligible levels after about 500 BCE. In contrast, populations in Castile, Catalonia, and the Ebro Basin show clear genetic signals of later admixture, with steppe ancestry fractions rising to 20% or more, and Mediterranean components increasing over time. The Basque gene pool, by comparison, stabilized at around 10% steppe ancestry, a figure nearly unchanged since the Iron Age. Mitochondrial DNA lineages, especially haplogroup H, show continuity from the Neolithic through the present, while the male lineages reflect the Bronze Age turnover. This combination of maternal stability and paternal replacement is preserved in unusually pure form among the Basques. Comparative analyses of gene flow rates confirm that the genetic distance between Basques and their nearest neighbors exceeds that found between many other European populations, separated by far greater distances. The evidence points to a scenario of isolation after admixture. Basque distinctiveness is not a relic of deep time separation, but the outcome of a demographic wall that formed after the Iron Age. The Basque communities themselves, through patterns of language, marriage, and inheritance, maintained this insulation while the rest of Iberia continued to mix. This revised model closes the debate over ancient origins and reframes Basque identity as a product of early admixture, followed by exceptional genetic continuity. Basque Y chromosome data reveal a striking pattern. Over 80% of Basque men today carry the R1b M269 lineage, a marker introduced during the Bronze Age steppe migrations. The same genetic signature dominates Western Europe, where it is closely associated with the spread of Indo-European languages. Yet, in the Basque valleys, the language remains Euskara, a non-Indo-European isolate with no known relatives. Genome-wide studies confirm that Basques share the same male lineage turnover as their neighbors. with indigenous paternal lines nearly erased and replaced by steppe-derived R1b. However, the expected linguistic shift never happened. The genetic evidence shows that while the Basques absorbed steppe ancestry, they did not adopt the language or culture that usually accompanied it elsewhere. Maternal lineages, in contrast, display deep continuity, with haplogroup H remaining dominant since the Neolithic. This creates a paradox. Basques are genetically shaped by the same Bronze Age upheaval that swept across Europe, yet their language survived unchanged. The presence of R1b without Indo-European speech demands an explanation beyond migration alone and raises the question of what cultural or social mechanisms preserved Euskara in the face of overwhelming genetic and demographic change. A sharp genetic boundary runs along the frontier where Euskara is spoken. Genome-wide studies from 2021 mapped a distinct allele frequency break at the language border, with the genetic shift often more abrupt than the physical landscape itself. Fine-scale analyses using over 629,000 markers revealed that Basque-speaking communities form tight genetic clusters, while nearby Spanish and French-speaking populations show clear signatures of recent admixture. Within the Basque region, genetic micro-isolates align closely with traditional dialect divisions. Parish marriage records from the Roncal Valley and neighboring Alava valleys report that more than 90% of unions between 1500 and 1800 occurred within the same dialect group. This pattern of intra-dialect marriage persisted for centuries, maintaining genetic separation even between villages separated by only a few kilometers. Spatial genetic mapping confirmed that the strongest breaks in identity by descent sharing coincide with dialect borders, not just with rivers or mountains. In some cases, the genetic distance between two Basque villages with different dialects rivals that between distant European populations. These findings demonstrate that language and dialect boundaries acted as effective barriers to gene flow, preserving both genetic and linguistic distinctiveness. The persistence of such barriers depended on community practices, marriage customs, inheritance patterns, and local governance that reinforced endogamy within linguistic lines. This microstructure created a mosaic of genetic continuity and separation, explaining how the Basque gene pool remained stable while the rest of Iberia changed around it. Basque inheritance customs shaped genetic boundaries that persist today. The ETXC system kept land and homes within a single family, usually passing to one heir. This discouraged marriages outside the community and limited the introduction of new genetic lines. Women held full property rights, with daughters inheriting and managing estates, which was an uncommon practice in medieval Europe. As a result, parish records from Roncal and Alava valleys show that over 90% of marriages from 1500 to 1800 occurred within the same dialect group. Mitochondrial DNA studies confirm this pattern. Bihar's 2012 analysis revealed Basque maternal lines have remained stable since the Neolithic, even as male lineages were nearly replaced during the Bronze Age. Flores-Bello's 2018 research on the Rh blood group adds further proof. Among more than 1,000 Basque individuals, 47.2% were Rh negative, all tied to a single deletion haplotype at the Rh-D locus, almost exclusive to the Basque valleys. These findings, property law, maternal DNA, and Rh genetics, together reveal a society shaped by isolation after admixture. Today, Basque genetic distinctiveness results from post-Iron Age isolation, not the absence of Bronze Age steppe ancestry. Genome-wide data confirm that Bronze Age migrations replaced most male lines, while maternal continuity remained. The old model no longer fits the evidence. As population genetics advances, identity and heritage debates will need to adapt. Origins are rarely simple. Preservation and change run side by side. Share your thoughts on what this means below.